So this is the last of the foundations for the chapter number 1. These are the genetic and evolutionary foundations. This is going to be the most brief of all the foundations, in part because these are courses that you could go on to take, in which you will get more in depth on these if you take if you take a genetics class or an evolutions course or an evolution course. So we'll be kind of brief with this. Life on Earth arose 3.5 to 3.8 billion years ago. The formation of self-replicating molecules was a key step in that, and we're going to talk about the experiment that was involved in developing that. Could it have been DNA? Could it have been proteins? That's kind of the question that was sought to be answered. And this was the prebiotic soup experiment, in which case a vacuum was produced, and I guess the conditions similar to that of the pre-life environment of Earth were basically made, where you had a lot of ammonia, methane, H2, hydrogen gas, water, and H2S, or hydrogen sulfide. So that was put under a vacuum. Electrodes were put in place there. A fairly hot environment ultimately led to the production of cyanic acid and amino acids. So this is kind of an early experiment where there's an idea that potentially from these simple molecules you could produce more complex molecules. And that was that gave rise to the theory of evolution. Now the question was what was the early molecules that were produced? Was it protein? Was it RNA? Was it DNA? And well, the theory is that there was an RNA world because of the fact that RNA can act as both an information carrier as well as a catalyst. There are some viruses that use RNA as a primary means of genetic information, so they don't have to go through the production of DNA. Now, sometimes those RNA viruses do produce DNA, so they can integrate that into a host genome and everything like that. But that's basically RNA functions to carry information and acts as a catalyst. So this is kind of the threshold, or this is kind of the progression of ideas. So prebiotic formation of simple compounds, including nucleotides from components of Earth's primitive atmosphere or gases in undersea volcanic vents, led to the production of short RNA or RNA-like molecules with random sequences. Now, those short sequences had selective replication or self-duplicating catalytic RNA segments, which gave rise to synthesis of specific peptides catalyzed by RNA, which led to the increasing role of peptides in RNA replication. So those things that were produced ultimately gave way to production of more of them, resulting in the coevolution of RNA and protein. Primitive translation systems developed with the RNA genome and RNA protein catalysts. That gave way to genomic RNA to be copied into a more stable molecule like DNA, resulting in a DNA genome translated on RNA protein complexes, the ribosome, with RNA and protein catalysts. So this is from that very early, or from that prebiotic soup experiment, the theorized progression onto DNA as our genetic store. Now, complementarity in DNA allows for replication with near-perfect fidelity. So this near-perfect fidelity, I think you can kind of read that as like, well, a perfect copying of it. And so what this shows is basically an originating strand up here, or an originating duplex of DNA that has two different strands labeled strand 1 and strand 2. Now those have reverse complementary sequences, which we'll get into more a little bit later when we're talking about DNA, but those serve as templates to make new strands off of one another. So from one molecule you go to two absolutely identical molecules. That gives way to the central dogma of biochemistry. I always was introduced to it as the central dogma of molecular biology, but it works for biochemistry as well. The idea was that DNA goes to RNA, which goes to protein. So as an example, you have a DNA molecule. Within that DNA molecule, you have a gene. In this case, we have the hexokinase gene. It's transcribed from DNA into a complementary messenger RNA strand. That messenger RNA strand goes to the ribosome, and on the ribosome is translated to a polypeptide chain. Now that polypeptide chain, that unfolded hexokinase, this is what we're looking at here, this is a protein that has not been folded properly, undergoes some sorts of contortions and movements, and then finally forms its catalytically active shape and structure. So that's kind of a quick view of what the central dogma of biochemistry or molecular biology is. Now, this is kind of our last foundation, evolution. Natural selection favors some mutations. Mutations are going to occur more or less randomly in DNA and in RNA. When they do occur, which if you take in a genetics class, you know about the different types of mutations, like a missense or a nonsense mutation. So even whenever you have a mutation occur, it's not necessarily guaranteed that it's going to result in a significant change in the protein sequence. So mutated polynucleotides may be transcribed and translated into molecular machinery like proteins. Now, mutations that give organisms an advantage in a given environment are more likely to be propagated. So what's the opposite of that? It's potential that the ones that give a disadvantage are less likely to be propagated, but something that is going to give an organism an advantage, well then that's going to be something that proceeds on through the generations. So natural selection is going to favor some. What we have here is a display of that. So we've got our hexokinase gene, and a rare mistake during DNA replication duplicates this hexokinase gene. So this is our replication event that has now we've got our original gene and our duplicate gene. A second rare mistake throughout this replication process results in a second hexokinase gene, and we have a mutation occurring right there. Okay, so we've got our original, and you could sometimes refer to this or look at this as a wild type, versus our mutant. So our mutant hexokinase. Now what this is able to do, mutant hexokinase with new substrate specificity for galactose. Okay, so the original hexokinase is going to take ATP and glucose and produce ADP and glucose 6-phosphate. ATP and galactose is going to, in our mutant form, is going to produce galactose 6-phosphate. Now, what's important to recognize about this is, well, and what's not really displayed in just this image is we've got two different sugars, glucose and galactose. Now those sugars are different from one another. This is an enzyme that is very selective, and it's going to go after only glucose. It's going to have a very low affinity for galactose. Our mutant is going to have a very low affinity for glucose. Now between those two sugars, glucose and galactose, glucose is much more common. It's much more accessible, and so if you provided a system with glucose over galactose, well, glucose goes through all these different steps in order to be metabolized and broken down completely down to CO2. Now, galactose goes through a couple of other steps, in which case it's almost like you're adding a level of complexity that didn't need to be there. So this would be something that would be a disadvantage. And as a disadvantage, well, this mutation is not going to be favored in subsequent generations. So evolution of eukaryotes can also be mediated through endosymbiosis. So in the event that you've taken cell biology class or something like that, endosymbiosis is something that you might be a little bit familiar with in the form of the chloroplast. Okay, so anaerobic metabolism is inefficient because fuel is not completely oxidized. So if you take, a bacterium, what is displayed here is we've got this ancestral anaerobic eukaryote. We've also got, kind of adjacent to it, this aerobic bacterium. This aerobic bacterium is very efficient because it's going to take any carbon-based fuel and break it all the way down to CO2. Okay, so what we have is this anaerobic eukaryote that takes on and that basically engulfs this bacterium. Okay, so we have the resulting aerobic eukaryote. Now this aerobic eukaryote is capable of then doing two things: anaerobic metabolism and aerobic metabolism. It uses that recently engulfed bacteria to go ahead and break down fuels all the way down to CO2. So this guy right here couldn't break things down all the way to CO2. This guy could. Now this resulting one can do that. Okay, so the result of this is a non-photosynthetic eukaryote. Okay, now if you combine that with a cyanobacterial genome or a cyanobacterial organism that is photosynthetic, so photosynthetic cyanobacterium, light energy is utilized to synthesize biomolecules from CO2. When that molecule gets engulfed, well, now all of a sudden we have both this this aerobic bacterium that has been engulfed, but now we have photosynthetic bacterium that has been engulfed. So the result is a photosynthetic eukaryote. And so that's basically the origin of your chloroplast as well as your mitochondria. The mitochondria was previously something that was all by itself, and cells engulfed them and it kind of integrated it as part of its repertoire of things that it was capable of doing. Now, when a cell took on a photosynthetic bacterium and was able to have an endosymbiotic relationship with them, now all of a sudden the cell is capable of breaking things all the way down to CO2, but it's also able to harvest light and produce sugars and produce sugars and go about its way generating energy from the sun. So that's our display of endosymbiosis. Now this wraps up chapter number 1. Lots of this was foundational material, and we're going to be revisiting them later on, but make sure that you appreciate this for what it is, and remember some of the details of all of this material. All right, well, I hope this was helpful. Have a good one.